It could be a silent mutation, it could be deleterious, it could be where the enhancers got removed, it could be the promoter got changed, something is not being transcribed, right? We're adding a lot more detail to it than just it's there or it isn't. Gene expression, excellent. Okay,
Can we repeat?
so I have blue eyes. What does that actually mean now? The proteins to make the brown eyes are not functional, therefore it was never made. Therefore I have blue eyes. It's not I have a blue pigment. That's not true. I am lacking the brown pigment.
Defective.
It's a defective pigment. Yes.
It is?
Absolutely. It's just that it was selected for in certain environments versus not. Okay?
Could you repeat, I'm sorry, the dominance phenotype means
Dominance is what shows up in the phenotype,
What
therefore
shows?
it's what protein is being transcribed, it's what's being expressed. So if we have something not being expressed, right, what does that mean? All right. What is a chromosome and a chromatid? And what is the differences? So talk, show what is a chromosome versus a chromatid.
I'm writing this down. Okay.
Your first step was replication. It began at G1. The initiator proteins bound. We had all of the DNA replication. You had all those enzymes involved, right? Leading, lagging, all those fun stuff. You ended up with two sister chromatids, which are just copies of DNA. If they undergo synapsing, now you have a parental and a recombinant. Now what happens? We undergo meiosis, right? What are the steps of meiotic division? So talk to you, what is meiotic division, meiosis, and mitosis?
Meiosis is two phases. Two phases. Meiosis 1 and 2. So.
But what is meiosis?
Mom, Dad, now we're going to go through every single step. We enter into G1. What got transcribed? What got transcribed when we enter G1? RNA of what? Of initiator proteins.
Initiator protein.
We transcribed initiator proteins. So we undergo replication because those initiator proteins bound at the points of origin, which opened up replication bubbles, fill in all those details. Now we have mom and dad. Okay, we've replicated. We have two sister chromatids. They come together and they synapse. We have our parentals and recombinants. We've swapped alleles. They go from to the metaphase plate, and the homologs separate. Okay, now we have two daughter cells. Those daughter cells are haploid. Excellent. Haploid replicated. And meiosis 2, the chromatids separate, and you end up with 4 daughter cells. All are haploid. That is where we are starting today. Today, now on, you now have all of the information and the biochemistry ready to build an animal. We're going to start building an animal. So we have now gone through cellular replication. We're going to now do it. So we're going to understand meiosis, and now we're going to start applying principles of actual making of the gametes. And so we're going to go through oogenesis and spermogenesis. Spermogenesis is the making of sperm. Oogenesis is the making of the oocyte, the egg. Spermicide. Yes?
There was a question on the quiz that I was confused.
We'll worry about that in a bit. Spermicide. I make enough sperm to repopulate the entire human race. Where you make enough eggs to have 1 or 2 children. What is the starting difference? From the reading, what was the starting difference? Talk to you.
resource. Todos como unas cosas. It was.
And remember what the difference was. It's the very first step in meiosis is different between men and women. No.
Prophase?
In women, we immediately go into meiosis. In men, we start with mitosis. So I'm going to start with a stem cell. That stem cell is going to duplicate. One stays a stem cell, one now becomes a spermicide. That spermicide then goes on through meiosis to make a sperm. But if I don't start with mitosis, I only have a few cells, and those turn into eggs. So if you were a female, how many eggs can you have? Many or few? Few. If you start with a stem cell and you make plenty more stem cells, over time you're going to end up with lots. So men will produce 300-400 million sperm in a day. Women are going to produce like 30,000 eggs over their lifetime. In fact, every single egg you've ever made was already made. All of your eggs were actually made while you were in utero, and your mom's genotype actually controlled that. We're going to talk about that later too. So, spermogenesis starts with a stem cell. And you might have an idea of what a stem cell is, but probably not overall. Cells come in different types. Toti means a cell can become anything. Totipotent means it can become anything. So toti means any. Pluri means many. Okay? So these are stem cells. Toti is it can become any cell at any stage. Pluri means it can become many different types of cells at different stages. Then we have what's called determined. It's going to become a few different types of things. A cell is determined to become a heart cell, determined to become a muscle cell. But are there different types of heart cells? Absolutely. So that is differentiated. Differentiated means one. Okay? So when we start with an oocyte, that egg is going to become any cell. So because we start with one cell, and we're going to make a whole organism. So that is a totipotent stem cell. As things turn on, we have different genes activated, right? If I need something on the 13th chromosome, we're going to turn those chromosomes off. The rest are going to become histinated. We're going to put them aside. Now it becomes pluripotent, means those are the things I can do. I'm going to become part of mesoderm, ectoderm, endoderm. What is ectoderm? Outside. What is endoderm?
Inside.
What is meso?
Medium.
Middle, right? There's your three pluripotents. We can become many things in mesoderm. I can become many things in ectoderm. But that's what I'm going to be. Ectoderm or mesoderm or endoderm. And then we're going to determine to become a tissue. A tissue is something like a heart, like muscles, right? Muscle tissue, nerve tissue. Those are the types of tissues we have. And those are going to become a few different things. And then we're going to be differentiated, means we are one thing. Okay? That's the only thing I can ever be, is the one thing. So in spermogenesis, I start with a stem cell. I can become anything. But I'm going to become a sperm cell. So I'm a determined cell. This is determined to become Sperm.
Sperm.
This is
The sperm.
the sperm. So that is called what kind of cell? Differentiated.
Differentiated.
That is a sperm cell. So the only thing a sperm can ever be is a
Sperm.
sperm cell. However, here I start with a cell, and it's going to become a whole damn organism.
Mm-hmm.
So that is totipotent.
Mm-hmm.
An egg is totipotent because it becomes
Everything.
anything, everything. Your cell, your egg, is everything. Okay? So that's our process, how we're gonna start. So today, we're starting with meiosis. We want to understand the principles of meiosis. Do you think Hardy Weber is coming back?
Yeah.
Yeah.
1000%, okay? And
Yeah. Eh?
next year gets worse. Yeah.
Ooh,
Instead
yeah.
of 3
Eh?
genes and 3 alleles, we're going to talk about like 40, right? And you got to learn how to model those things. So
Great.
we're going to start with the sperm. Sperm are the simplest, stupidest things in the world. We have a head, neck, midpiece, tail. My first question for you.
Oh wait, what happened? Why am I not doing? Thank you.
Can you pass one of those worksheets to all of us?
Oh, I didn't know I had all of them. Sorry.
All right. 10 seconds.
Wait, we don't have a question open.
Yeah, something weird happened.
Oh, I need to refresh this.
All right. So it looks like we're struggling with reading comprehension. Figure it out.
Oh wait, what was it? Where are the most mitochondria found?
What are the most mitochondria found in the sperm?
Oh, in the nucleus?
Well,
Oh, it doesn't say.
use insects.
Meat piece.
Lots of them.
Oh, mitochondria there. Central. In the central? Or is that the? Oh, I put central and flagellum. What did you put?
Yeah. Oh, there. Yeah, that's right. Yeah, it's in the middle.
Where's the meat piece? Oh, yeah, yeah. Yeah, yeah.
Flagellum is a part of
hungry.
it.
All of a sudden we can read now? Oh! We're now learning to read all of a sudden. Where are the mitochondria located?
No, not
Meat piece.
The mid piece. Come on!
Oh, midpiece, okay.
They're all right there.
I was looking at this side and
All right.
I was
So this is a
listening.
diagram. This is what they look like in real life. Tell me something interesting based on in the mid piece. So how do sperm actually work? From the reading, how does sperm work? They swim. Cool. So the flagella beats, okay? And eggs produce a chemical, a signal, and the more it detects that signal, the faster the flagella beats, therefore it'll find the egg. If it's detecting less of the signal, the flagella beats less, so it won't swim away, it'll swim towards it. It has things like an acrosome at the front, which was digestive enzymes, which are going to help get into the actual egg. And then it has the nucleus. The nucleus is attached in here. What does the sperm actually inject into the egg? Only the nucleus. That's it. So men only give the genetics for the nucleus. They don't provide anything else. They provide no nutrients, they don't provide any mitochondria, they don't provide anything else. So, where did you get your mitochondria from? Your mom or your dad? Mom. That's interesting, okay? So, you only get that from mom. And where did you get all of your organelles from? Mom. All the genetics, and all the capacity, the building of the proteins, the ribosomes, you get all that from mom. That's because of the shape and the structure of the mitochondria. Or, sorry, okay? So let's get started on the worksheet.
You're an unreplicated condensed chromosome. Circle a homologous pair in the karyotype. Remember one of these chromosomes came from the male and the other from the female. These two carry the same gene. Circle a homologous pair. For this organism, its chromosome numbers 2 identicals. One, 2, 3, 4, 5, 6. 2 equals 6. Label each cell in Model 1 as either diploid or haploid. Label each cell in Model 1 as having either replicated or unreplicated. What do homologs during meiosis that does not occur do during meiosis that does not occur during mitosis? Final products of mitosis and meiosis.
Isn't it replicated all the way to the bottom? Or no? Yeah, so that's not replicated. This one's all replicated.
Mol 2. Mol 2. Does meiosis or mitosis occur between age 1 and 2?
Oh, oh, yeah, they're not, so we consider the mitosis, the homologs are replicated. When they split, right? That one replicated, and then meiosis are unreplicated when they split. And then that one's unreplicated. I said it for 3, I was thinking 4 dollars.
On the chromosomes in their proper states, in the unfilled cells in panel 7 to 9.
No.
That's funny.
they're haploid. And there's 2 daughter cells versus 4.
Yeah, I'm saying mitosis is diploid and haploid, I mean because I know it's like that was actually so much fun when we got like right chalk. There's probably a lot No, wait, my answer is next. Yeah, it's next week. What are we doing?
Testicles and
That's so much fun. From humans?
No, it's actually not. It is, because we're learning the structures. You're learning all the structures, you're doing the techniques, and the animals aren't dead. It's just from staying. So you're learning everything, you didn't have to kill anything. So it's awesome. It's an absolute awesome life. You should. Except that you don't typically neuter them. Now, would you spay women? That is much more common. That's called hysterectomy. Well, no, because you don't want to remove, you live longer, so you don't want to remove something. And dogs don't live long enough, so the hormone removal is. The second is that's why. The second is usually because the second is too good, and you're finding it. And that's the same as if you're too tired. Right? Hysterectomies are when you remove all structures. Castration is when you remove the testicles.
Yeah, see, I freaking, you just fist bumped me. You guys promote you as the one. You can't, it just happened.
I'm going back home.
It's just awesome. Sorry. Oh my God, my dad has a dead brother, and I forgot. So I went, I'm so sorry your brother died. I'm getting 5 big boobs, and I paused over and
Alright. So let's walk through this real quick. In mitosis, I start with a cell and I end up with 2 daughter cells. Are these daughter cells identical to that?
No.
Yes.
Oh, yes.
Because this is just replicated, right?
Oh, yeah.
So I am 2n equals
6.
6. Mom, dad. Mom, dad, right?
Yep.
2n equals 6. So they're replicated, they line up, and then the sister chromatids separate, go to the poles, we get cytokinesis and cleavage, and we cut the 2 cells. Right? Meiosis, we start with the same thing.
Same thing.
Mom, dad, we replicate.
We
Same thing.
replicate.
But instead of lining up, we synapse, where we can get crossing over, which is recombination, and then the sister homologs separate, go to the poles. Now I am haploid or diploid?
Haploid.
I am haploid, replicated or unreplicated?
Haploid replicated.
Replicated. And so now the sister chromatids get pulled apart, and I end up with how many daughter cells? 4.
Haploid
Same or different? Different. 4 daughter cells, all different. All right, keep going. We should be on model 2 now.
I'm halfway through.
didn't look almost separate.
Spermatocyte, primary spermatocyte, goes from meiosis 1.
It's still meiosis. Not in the compound. But. Oh, yeah. Wait. In your circle.
Starts over again. That's the stem cell. Correct. Because
That's a big part, but it goes back to what you're saying. And then it goes back to the, like it's starting.
good.
Yeah, because I've seen now. Wait.
Spermatocyte is completed meiosis.
Yes. Like, yeah. It goes to 42. I'll take a nap right here.
This is meiosis. This is meiosis.
Spermatozoa.
And why
So let's look at panel 1 to panel 2. What is happening? Mitosis or meiosis?
Mitosis.
Mitosis. Now this is cool. So we start with the stem cell. And what type of stem cell is that going to be? It's a determined sperm cell, right? So it's a stem cell. It's determined to become that. Is it differentiated to become sperm yet?
No.
No, but it's pretty sure it's going to become sperm. So it's a determined cell.
Oh.
And it's going through mitosis. Then we have two new cells. This one is going to do what? Go through meiosis. Excellent. But this original one is going to do what? Mitosis again. So as long as I'm doing mitotic division, I can produce endless number of sperm. How many sperm does a man produce? Endless.
A lot.
Over the lifetime, maybe 1 trillion. More, maybe more over the lifetime. Hell, actually, let's do that math. 300 million a day. Every 3 days is 1 billion, so 3,000 days is 1 trillion. Yeah, you're starting to get into trillions of sperm cells. All right, so over, say the average man lives 75 years, which is pretty low. They're making sperm from 12 to 75. Yeah, we're in 10 trillion or so. It's a lot of sperm. That's because we're doing mitosis. Okay? Keep going. Get back in there.
Suggest a reason why both the daughter cells in the division in panels 1 and 2 do not go through the same process.
I don't know actually. It's like we're in the test stage of the exam, and then it's like
Yeah, but that's because I think like it drops down from like the mid there drop down and then it goes out of that.
I have no idea.
Oh, it is. You're right. Yeah, wait, what is, yeah. But split, so would it be one of
2 to 7.
What?
From 2 to 7, they go through what?
From
Oh, that's
2 to
number
7
10.
they go through meiosis.
Okay, that's what I. Oh, 7 to 9. Oh.
Okay. So my one is That's not getting there. I feel like that's really a final.
Like 2 weeks. What is that? Okay, good. I know. I think about whatever. Basic, let's go with basic. Early life, there's their accomplishments, I don't know.
The struggles and what they're known for.
You got it. Wait, what's the first thing?
Oh.
So we're going to like.
Alright, let's go.
We need to know.
Oh god, there's an arm.
I feel like that makes sense if it's starting.
Rather than starting at step one.
I was like, guess all we had is vacation.
Your head circle is just slightly bigger.
Oh. Scare me. No, you're good.
Bless you.
Bless you.
My number is but then Brian should start with my advice
Oh, wait. Meiosis.
Wait, oh, okay, so it's like, this is like a section within there?
Yeah.
Okay.
Model 3. How does the number of oocytes change when you say cell?
No, but I mean, it makes sense that I'm like, this is but so then I'm not insane. No, I haven't.
What would you say is meiosis two?
What are you trying to do? Model 2. Oh, Model 2 Part 1.
In the 12. The number 12.
And number 12. I labeled everything the best I could. I'm not sure if it's right, but this is what I labeled.
Where would you
This is how I did it.
Quizás this is first and then second.
Primary sperm up. Whatever it is, because I don't have the paper, I don't know how to say it.
Okay, thank you.
Spermatocyte.
Thank you.
Spermatocyte.
Because now it's meiosis.
Shows you the help. What's the help answer the question I was trying to figure out? I guess it's 18.
Oh.
A Model 3 question.
I don't know.
This can't be the larger cell because it's.
Doubles? No.
Okay, let's see.
What happened to the other one? I don't know. I don't know what it is.
Like one of them is like good than the other.
Yeah. Oh, really?
It's made in testing. The first one be seven.
This corner, got it.
Okay, so let's get knowledge to throw it out. Thank you.
Oh, you know what?
Yeah, it's the
In
seven.
Carolyn, there are two types of cells shown in the follicle.
Oh, okay.
Are the follicles going through my system? Maybe the drawing is really just growing.
Yeah, I think follicles are nice, but
Yeah.
I didn't know where they are.
How is it ever
So I guess
oocytes change over the summer?
the reason why like those stock miners for resource opportunities to grow the index.
Okay, so we're done.
Yeah. Oh, this is for
Yeah. Because in the offsite.
I remember that from reading like last Friday. Yeah. Okay.
Like the egg can stay alive. Okay.
16 isn't even a question.
What type of cell division is the? Oh, I see.
Oh, I didn't wait. One large one surrounded by. And that is not yeah, because we're on
You're doing so good until freaking Model 3.
Sorry about that, guys.
You're fine.
When it rains, it pours. Now we found out one of our faculty members won't be here for spring, so now Jeff and I are juggling. How many classes can I take? So that'll be fun. Okay, so we have a stem cell. Now we've gone through meiosis.
You're good.
This is a determined cell. We're determined to become sperm. Have we become sperm yet? No. This is where we become sperm. Okay? So this is a differentiated cell. So we've completed meiosis. At that point, this cell is going to go through mitosis again. So we're restart the process. So we have a spermatocyte, and then we have a spermatid, and then we have sperm. Okay? Where they're going to mature to become. Have we completed model 2? Yes. We started model 3. Did you get to model 3? I have a question.
Yippee. What did you answer for number 13? On model 2 part 1.
13?
You just show the chromosomes. That nothing has to be wrong.
Nothing.
Just the restarting.
Yet.
Correct.
You saw your
Because it's a
question, right?
cycle, just continuously.
What?
You saw the question?
Oh, the question? Oh, there's a multiple. Oh, okay.
I don't know what
The topic is undergoing. So.
So, it's 3 times here. And then 3, 2. I got it.
So then for these questions. Well, this is smart.
I didn't know if it was that or like the next one you put in.
No, just actual.
Okay.
They're gonna be counted around 635.
Yeah, so that's the second one.
Because you will, by the time you submit your Western application, I think winter quarter is done.
Yeah, because 8's the first one, and then 9 is the first.
About 35 miles south. I'm the CA. Do you think I would read in those worksheets for you guys or the same one as one?
Same one.
Because I am lazy. I wanted to hear, I mean, I want to leave. I can't. Yeah, that's what they're PhDs. No. Yeah. You get to be a PhD. That's what teachers are, PhD students. They're learning to teach, they're learning to fix. Yeah, I would love to figure out things, but I don't have to grade.
Polycrystal surrounding the oocyte transport protein mRNA nutrients into the oocyte, which in turn grows larger. Suggest a reason for this intensive stockpiling of resources on in the oocyte.
That is the benefit for faculty. I teach way less.
Well, it's basically asking. So.
All right. So sperm is produced in what location?
The epididymis.
Testis.
Seminiferous tubules.
The
The seminiferous tubules are where sperm are produced.
seminiferous tubules.
Okay? Then they're going to go through the vas deferens to the epididymis, where they're going to hang out and mature. Then, when they're released, ejaculation takes place. They're going to move through the urethra out of the animal. Okay? That is how we're conducted. So, the correct answer is Two, sorry, 2.
3241?
Yes, 3241.
I got it.
Okay? So, next week in lab, we're going to be dissecting testicles and ovaries. Okay, it's your first actual dissection. It's a really cool lab. Ovaries are very boring to look at until they are not. Okay? Most of the time, the animals that we get them from, ovaries are pretty immature. So, they're just boring. Every once in a while, we have an animal that was pregnant. And you will see like the blastulas and the gastrulation taking place in there. And that's cool. There's not going to be any little puppies or kitties inside of them, okay? It's going to be like little balls of cells. The testicles, though, the seminiferous tubules you can see, and you can get, you can look at sperm, and we'll be able to look at all that. The seminiferous tubules, the best way to describe it is if you have a pile of cooked spaghetti just sitting there. Yeah, that's the best way to describe it. There's just lots of tubes everywhere, and that is because we want to increase our surface area. Sperm, each cell is going to produce 4 sperm cells. So if I have, it is in a tube, and lots of these tubes, I can produce millions and millions of sperm cells at any given time. Then they go to the epidermis where they hang out and mature. Okay? Should be on module 4 now. Model 4 or 3, sorry. Model 3.
Yeah, I think that's just my I thought you meant like put no on one.
That's the O side, is the middle. So,
Yeah, it
is it
just
every cardinal system? No. So stays the same.
How did you answer number 19?
Just a
Stuck
second.
on that one.
Oh, you already wrote that. Okay, you're right.
19?
Yeah.
The follicles surrounding
Because that's why like only like
in turn grows larger. Suggests a reason for this is a stock phytoplansm. They stock to prepare for development early of the embryo and for preparing for the initial stage of growth and division and fertilization or something.
When does meiosis 2 happen? It was in the reading.
Oh, isn't it?
After fertilization.
Probably none of you have completed meiosis 2.
Right, so the oocyte has to provide
Right.
yeah.
the organelles.
Huh?
Damn.
Oh, I was like, what did you say?
All start working together. I'm so proud.
Okay. Yeah.
Right, but I wasn't very different.
Okay, wait. Finish. What? That's not it. Okay, what did you say so far?
Alright, so the sperm goes through my testes.
Yeah.
What?
One daughter cell.
starting at panel 4, fluid was being released into increase the size of the follicle. Which type of cell is most likely responsible for the release of this fluid?
Follicle cells.
Follicle cells?
What did you put on 20?
Yeah, I haven't been on 20.
Oh, okay.
Panel 7 shows ovulation. Has the released oocyte repeated?
No. Morning sickness has to do with, there's some thoughts, brain development, neurulation. Long story short, that's what it's thought. The other aspect is it could be regulation of certain genes. Morning sickness is far more likely in a female. So if you have severe morning sickness, you're much more likely to be pregnant with a female than a male.
My girlfriend has sexual emergency, so they said it was a girl.
When did you find out?
So much more likely to experience morning sickness. There is some accuracy, but a lot of people, I've heard always said it was males being born with morning sickness. But okay, well, then that is much more likely. And we will cover probably the reasoning why, and like next week we'll talk about neurulation and differentiation.
I'm just curious because I see so many this is why.
You know, it's most likely due to gene expression. Long story short, mom's genotype actually controls the first stages of development. And so, like, if something went wrong in pregnancy, it was because of mom's genotype, not necessarily the embryo's genotype. And so, mom is trying to compensate for other things when it's a female because now mom's getting estradiol from the embryo and estradiol from her. And there's a compounding estradiol level, so that would cause increased morning sickness. We'll talk a lot about development over the next 2 weeks, and then we're going to cycle back to it and not upon when we do menstruation.
Spermatogenesis is the released oocyte a one primary or
Majority
secondary?
of your women, it's nice to know about your own body. Yeah, that's unfortunate.
Women figure it out and worry about the guys.
The truth about their bodies, and when we stigmatize, we make things seem like it's off limits. Sorry.
It's awkward and non-effective.
You are a woman. You have female parts. Those experience certain things.
It's more destructive.
Yeah, absolutely. Like, and if you want to stop children from having sex, I got a great way to stop them from having sex. You teach them what happens. Yeah. You sugarcoat things, but you know, it's like here, daring me to do drugs, okay? I'm going to do the drugs. It's much more likely curiosity. I teach you about diseases and what actually happens when one's at least like, oh, hell no. Right?
Exactly.
Let him take.
Education.
Let him take care of a baby for a few days.
Let him have Sylvie last night, okay? She had her first night terror ever. It was sad, but it took her about 45 minutes to calm down and recover. And that was the worst 45 minutes. Screaming. And she never has that problem, but like, she had it. But Charlotte and I, we worked pretty well as a team, like we handled it pretty well. We decided we're bringing earplugs into the bedroom in case that happens. Our bedroom's echoey. So all right. What's the big difference between meiosis taking place in sperm versus eggs? First step, what's the big difference?
Eggs go to meiosis.
We immediately go to meiosis. And we enter in meiosis 2, and what happens? Or sorry, meiosis 1.
Oh, Mark.
What happens? You stay there. We are frozen. This is a primary oocyte. When does meiosis 2 complete? When you get pregnant. At fertilization, once the egg penetrates once the sperm penetrates the egg, it'll immediately eject the second polar body and complete meiosis 2. So meiosis is held. You are held in G2 checkpoint while your eggs are being in your body. So every woman in here, every one of your eggs was made while you were still an embryo. While you were a fetus in your mom's womb, your eggs were built. All those eggs are frozen in time in meiosis 2. And what happens is we undergo follicular development. And the follicles get produced and they start getting bigger. And essentially this is a pimple. Okay? So I need to squeeze out and push out the egg. And so we're going to expand those, but at the same time, I'm going to start transferring nutrients to the egg because that egg cell needs to get really big. Our eggs are very, very tiny, okay? I mean, they're itty bitty. You can see them on 5 and 10x under a microscope, but they're pretty small. Eggs can range in size from that to literally a giant ass egg. Okay, that's one cell. So I need to put all of that nutrients in there. At the same time, I got to squeeze it out of the ovary. And so those follicle cells are going to engorge and push it out. It's going to then enter into the fallopian tube, where it will either meet a sperm or it will not. If it meets a sperm, we undergo fertilization, and then we will experience, we'll talk about that here in a minute, a calcium wave and then a jelly-coat formation. And then we'll undergo cleavage. We'll become a zygote. If it does not meet sperm, it will pass through. It'll pass through the fallopian tubes and out the vagina. It will then trigger, because you have not embedded into the uterine lining, it will trigger menses, menstruation. If you are pregnant and the sperm meets the egg, it will embed into the uterine lining and create a placenta, which will not trigger menses. Okay, that is how that takes place. So all these follicle cells are producing lots of nutrients and pushing the oocyte out.
So what's happening in people who menstruate while they're pregnant?
That's actually not true. Okay, so you're not menstruating. Now you can have bleeding and other things that is independent of menstruation. Because if you have menstruation, you shed your uterine lining. The hormones in there will cause the uterine lining to shed, would disconnect any egg there, and it would be a miscarriage. Okay, so you can have bleeding, you can have other things. There's all kinds of things, preeclampsia to hypertension, diabetes can cause that. So there's lots of other factors that can come into play. Now, yeah.
So if there's no fertilization, then it never becomes a secondary oocyte?
Correct. Yeah. Now, does anybody know what cystic ovarian syndrome, polycystic ovarian syndrome is? Or what that is? Anybody have experience? I don't want to throw people under the bus, but you've experienced an ovarian cyst?
Yes.
Yeah, you've experienced an ovarian cyst. Most women, unfortunately, will experience one. Okay, they're awful, okay? So essentially what happens is these follicle cells will engorge and expand and there's no stopping it until it bursts open. And they can range in size to a little one, like just a little bit more than a normal oocyte, to as large as like a grapefruit small watermelon. And that's inside you. And so when it bursts, it releases fluid into your abdomen. Is there any blood and fluid in your abdomen normally? No. You are not an open circulatory system. You have a closed circulatory system. So contrary to popular belief, you don't have blood swishing around in your abdomen and around your organs. There's no such thing. Okay, you are a closed circulatory system, means everything is in vascular tissue. So when that fluid bursts and expands, because these follicle cells have engorged too much, there hasn't been a trigger to stop it, that releases the fluid in the abdomen. And you have receptors in your abdomen that say, is there fluid here? And what that does is it gives a pain response. And when you have fluid in your abdomen, all of a sudden now you have fluid there, your pain receptors go off. And my understanding, and I don't want to speak for a woman, is it can range to like extreme pain. Like a 10 out of 10. It's as bad as it can possibly be. Polycystic syndrome is you experience many of these over your lifetime. Most women will experience one. It's just random chance they'll experience one. Ironically, the best way not to experience them is to have children and many children. The second, so ask me, put your thinking hat on, why? Now talk to each other. Why? Why is that the right answer? What is stopping? Or what is not happening when you have many children?
You're just going to have less menstruations. It's like normally it's 28 days, but when you're going to get pregnant, it's minus 9 months. Not like 2 months after the pregnancy, so you're just going to have the less possibility to happen.
Sounds pretty accurate. I would say it's logical. Sounds logical.
I don't know.
All right. Why? Why is that a good way not to experience or to stop them from happening? They got an idea? So if you want to stop ovarian cysts from happening, you just have many children. Why? You're not having oocyte development. When you are pregnant and not undergoing menstruations, you never release an egg. You don't go through oocytogenesis. So if you, while you're pregnant, and then often while you're lactating, that is stopped. You not typically go through oocytogenesis. So we're not going through production of an egg. So it just stops. Now, the question becomes, how can you do that without being pregnant? Birth control. Excellent. So there are 2 types of birth control. There is one type where you can stop eggs from being released, okay? So you will never ovulate. That is one type of birth control. The second type of birth control is where you ovulate. You can, in fact, get pregnant, and often do get pregnant, but the egg can never embed in the uterine lining. Therefore, when you menstruate, it just goes away. Those are the 2 types you can typically have.
Why would you want to keep your period?
So then the question becomes, why keep your menstruation? Is menstruation good for your bones? No.
It's pretty
It's
bad.
actually really bad for your bones. So, do we talk about what bones are for? Oh, we're going to go down deviation. Now we got to go down deviation. Okay. Does anybody know what bones are actually for? The original evolutionary purpose of bones.
Oh, you did tell us. I can't remember.
It's not protection, not
It
structure,
wasn't structure.
not for movement. It is made out of calcium phosphate. It's an energy reserve. calcium phosphate, calcium to move your muscles around, muscles sliding, actin filament sliding, and phosphate, adenine triphosphate. So if you are low on energy reserves, what happens to your bones? They get taken away. What happens to women over their lifetime? They lose their bones because of your menstruation and those parasites that you end up having. Okay? All right? They're literal parasites. So they suck your bones away.
Wait, who's the parasites?
Children.
Oh.
Those leeches inside.
I should have known. They suck up everything.
Then the question became, you're on birth control. Why did you keep your menstruational cycle? There's no reason to. There's in fact no medical or biological reason to do it. The only reason to do it is often they found when they were marketing it, women wanted peace of mind. So they put 4 or 5 placebo pills in there so you'd get a spike in your hormones to trigger a menstruation once a month. In fact, you do not need to have it. There's no medical reasoning to have it. So if you do not want to menstruate, talk to your doctor and go. You can probably just get rid of the sugar pills and keep going. So actually there's a lot of evidence to say that if you do not menstruate, your chances of ovarian cystic syndrome decreases, endometriosis decreases, and your bone density increases, and your life expectancy increases. So talk to your doctor about that.
But if they take your matrix out.
Yeah, that's a hysterectomy. That's totally different.
Now, if
we have a lot of women in here who are like, I'm making a doctor's appointment today. Go for it. Yeah, menopause, where you stop menstruating, is essentially you have ran out of eggs, or that your system can no longer tolerate the extremes up and downs of hormones. So you basically fade out. It fades out.
So, if our evolutionary purpose is to create life, why do we stay alive after?
Look, great question. You shouldn't.
So why do we?
Why do we? Why do nautiluses? Does it matter? Maybe it doesn't. Maybe it's just an accident. Maybe Rick and Morty's right and nothing matters, right? What if it does? I don't have that answer. Right? So evolution said as long as your children are old enough to go reproduce, you shouldn't matter anymore. So that's a question to think about. That's a philosophical question that people debate all the time.
So when, if you would do that and stop menstruating, can you have children still?
Yes, all you'd have to do is restart.
Just restart. Okay.
Stop the hormone therapy. Does anyone know the history of birth control? History of birth control is actually really fascinating. Many cultures around the world have had birth controls, actually very effective birth controls. Not funny. There was a plant in the Mediterranean that was exceptionally effective birth control, probably way more effective than ours currently today. And it got used to extinction because women didn't want to have as many offspring. So during the Romans literally harvested that plant to extinction. But there was, in the 1930s, there was some information being leaked out of Mexico. And people said, if you eat this yam, you will not get pregnant. And everybody thinks that's full of bullshit, right? Just not correct. Until a doctor from New York went down and started doing a study. And he found out that if women actually ate this yam, they do not get pregnant. Turns out the yam was full of estradiols and progesterones. And that is where the discovery of birth control came from, the modern birth control. So that's how we do it today. We control it with the progesterones. Essentially, we're going to learn about that next quarter when we undergo tissues and start learning about menstruation and how the tissues actually work. But if you regulate that level and you no longer have the spikes and draws, and just keep that, your body thinks essentially you're pregnant and will not form and pregnancy. It's a fascinating subject. Okay.
Very fascinating indeed.
Let's finish up the worksheet. We'll get started here on the next question in just a second.
I was going to ask about the primary.
It makes me dizzy having to flip back and forth. I don't know why, but it makes my brain. And I'm having to watch.
Yeah, I think this is like some of the
Did you happen to answer any of these questions?
The meiosis is produced, or will the polar body survive? It says, nope, it wouldn't survive. Is that what you have?
Yeah, I said that.
Compare this nitrogen to the rest of the secondary oxytide. I mean oocyte. How do you
It's primary. It's primary.
primary?
Yes.
But, I know, but it's due today. I
Spermatogenesis involved.
I mean, but after today. My parents are gone and I don't know. I have to remind myself I have to go and see. Like, they want their computers turned on. No, it's on the computer, so I have to like log in and kind of work their files. You know, I'll become like a computer scientist. I'll hack into their system. I'll be like, guys, we're Is it for
Relates to diversity or
Okay, so did you get for 17 follicle cells, or what did you get?
20.
17.
Follicle cells increases if you look at all the panels, I think.
That's what I have.
No, it stays the same.
Oxides remain the same and follicles increase, right? Is that what you have?
Follicles stay the same, but oocytes increase.
Oh, and 20, do you have follicle cells?
Yeah.
I'm sorry.
Which question you were asking?
Oh yeah, stay the same, increase. Yeah, and then do you have which cell? Follicle cells? Yeah. Oh, and then you have yes, no,
Yeah, no.
release oxide completed meiosis 1, but not meiosis 2. That's what I have on this. I need to ask this one.
What did you get for 21C?
I think we have a question. I'll get you. Okay. We're asking.
Yeah, 10 more seconds.
21.
21C.
2 looks like that's what, but he says for, so I need to ask that one too.
All right? Not doing so good. Talk to me.
I'm not sure. No, I think
I picked distribution of cytoplasm.
second one when you get pregnant.
Oh. I think you're right. I'll ask.
Like it's staying like in a board between 1 and 2, but it's getting to 2 when you get pregnant. But I'm not sure about it.
What did you do?
I picked A.
I did too. No, I picked B.
I think it's because the teacher has questions back here.
Is this true? No, it's equal. That's incorrect. Primary spermatocytes undergo meiosis I to produce diploid secondary spermatocytes.
No.
No, they're haploid. Only one of the meiosis I products of primary spermatocyte will mature into a sperm.
No.
No, all 4 mature. Oh my god. Primary oocyte production stops in development in many mammals. Yes, including humans. It's all frozen in you until fertilization. So 4 is the correct answer.
I picked it first, but then I picked 1.
Let's imagine copulation has occurred. Normally, in the past, we used to do this lab. You're going to do this online. Okay? So before the end of the quarter, you have 2 labs you need to complete. They're both online. One is sea urchin fertilization. Sea urchin fertilization actually in lab is really cool to do. The problem is there's almost no sea urchins left in the wild, so to get them is really, really hard anymore. But sea urchins produce really big eggs. And so what we do is, sea urchins are male or female, and you would extract sperm or eggs from them. You would stick the egg under the microscope, and then you would take a dropper with sperm and drop it on. You would then watch the sperm swim to the egg, where then it would undergo and find the jelly coat. The acrosome would be released, and you'd be able to see this. And the jelly coat would be dissolved until the sperm penetrates. Once the sperm penetrates, it would undergo a calcium wave, which then would cover the coat of the egg, breaking up all the receptors, and the jelly coat layer would expand. This is what it would look like in lab.
I'm gonna watch that video.
Okay, so this is what you would see. So it happened really quick. So we'll restart there, because then we'll talk about this here in a second. All right, so sperm gets inserted into it, it fertilized, the calcium wave covers it, and the jelly coat layer expands. That jelly coat pushed away the sperm. Okay, so we'll do that again, just really quick. Okay, so sperm. So you'd be able to see this in lab. Then what is cool is they undergo meiotic division. So you'd watch the polar body be expelled, and then this is meiotic division. So we've created 2 cells, and then we'll create 4, and then we'll create 8, 16. Meiotic division in sea urchins takes place about every 50 minutes. So you would have been able to watch 2 cleavages form. Okay? So in lab, you would have seen fertilization. Okay, so this is fertilization. Then you would see blastulization. We're creating a blastula of all totipotent cells. All these cells become something. They'll become anything. And they'll undergo until you form for about 48 hours of blastulization. So double every 50 minutes, that's a lot of cells. And then this is called gastrulation. Gastrulation is where we form our 3 tissue layers. They go from totipotent to pluripotent. And then the organogenesis, so we start developing into those tissue layers to actually become something. And then we would go what was called neurulation at that point. Neurulation is where you start forming your neural tubes, and then we grow out to become an organism. In lab, you would have seen the first 2 cell divisions. So online, you're going to do this and actually watch it and do some of the experiments. Okay? So there's that. So what happens? Right? So sperm receives a signal That signal tells the sperm to swim faster. It's going to release, enter the jelly coat. It's going to release the acrosome, which is going to be a bunch of enzymes. It's going to help dissolve and be able to penetrate. Once it penetrates, it's going to release its nucleus into the egg. The egg then is going to undergo a calcium wave. That calcium wave is going to encompass the cell and basically break all the receptors off so that it can no longer receive any additional sperm. And then then the jelly coat layer is going to expand to push everything else away and to protect the oocyte. Protect the egg.
Is it possible for an egg to be fertilized by two different cells? Sperm cells?
Excellent. So let's imagine that happens. What would we call that? What? It's not. Because what would the ploidy be? 3. So what would happen? It would die. The checkpoints would hit and it would go on. In humans, do the majority of fertilization events turn into be a successful offspring or not? No. In fact, most fertilizations end in abortion. Not talking about artificial where you choose to have an abortion. I mean natural abortion. So not even necessarily a miscarriage. You would never even know you're pregnant. So something went wrong in the first 48 hours, 72 hours. It just doesn't progress. So it's very, very common. Often it's something really simply stupid like an extra chromosome or a chromosome misaligned on the first meiotic division. It didn't line up correctly. Boom, done. It's over with. A chromosome's microtubule is off. Boom, done, over with. The jelly coat layer formed in an abnormal way. Over with. So all these things have triggers, and if any trigger takes place, it ends.
What's the reason for a chicken egg having 2 yolks?
Excellent. It didn't. They stuck together. It went through. It didn't unbind. So my question then becomes, how do twins work? No, it's nothing to do with polar bodies.
Isn't it 2 ovaries?
Yes, excellent. Fraternal twins are the female released 2 eggs. Not uncommon. When you think about a dog, they have a litter. How many eggs did they release? 6, 8, 10. Release lots of eggs. Humans release typically 1 egg. Every once in a while they release 2 eggs. So in the case you say, oh, twins run in my family. Usually that's on the mom's side, and it's fraternal twins. The second way it can happen is, let's imagine during blastulation, the egg broke in half. Okay? It unheased. The cohesion accidentally happened. That would be identical twins. We have the same genotype, the eggs broke in half. So those are your 2 ways. Okay? We'll finish up the worksheet. Go finish that up on your own. On Monday, we will begin with neurulation. I will see you all in lab tomorrow, where we're going to do respiration.
Tomorrow lab.
Yep, tomorrow lab.